morning, everyone. My name is Ning Xiao. Sorry, my name disappeared from the program. But anyway, I'm a PhD student at University of Cambridge. I work closely with Krishna Mapadabi, which is my secondary supervisor. And then my primary supervisor is Professor Nigel Slater, which is the head of the department at the moment and going to be the Pro-Vice Chancellor next year. So that's why he's very pushing up and try to make me finish the PhD as soon as possible. Anyway, I do realize it's about lunchtime, so I make my presentation as short as possible. And then it's very preliminary results I've got here because of the short notice of the presentation. But if you have any questions, you are more than welcome to ask me after the presentation or during the Q&A session. Anyway, today I'm going to talk about using a photosynthetic bacterium as a biocatalyst for hydrogen production, which is very close to biorefinery research as Professor or Dr. Christopher is doing. So hydrogen, a new green sustainable energy, can be generated from the photosynthetic bacterium under the nitrogen fixation process in the anaerobic condition. From this simplified mechanism diagram, I make it much easier to understand, and we can see at least 2 points. From the energy side of it, if the light is shined on the bacteria, it will be absorbed at a reaction center and converted to ATP energy carrier and supply the energy for both cell growth as well as hydrogen production. From the material side, if you supply the organic waste, then it can be utilized by the bacteria as a source of carbon for cell growth, also as a source of electrons for hydrogen production. Therefore, both hydrogen production and cell growth will consume energy and organic substrate. Normally, people can use a lot of different organic waste in industry as organic substrate, such as glycerol from the biodiesel industry, or any other biomass from the waste food industry, or from any other biorefinery downstream. We select a very specific, unique strain of bacteria, which is called Rhodopseudomonas plastris. It is a very common soil and water bacterium, used to be widely applied in the wastewater treatment, and it is very metabolically diverse. But the advantage of this specific strain of bacteria is that it does not produce any oxygen during the whole photosynthetic process, which is very good for hydrogen, considering hydrogen meet with oxygen, they simply just combustion. And here is a very simplified schematic diagram of the experimental setup. We use incandescent light bulb to provide the light for the bacteria. The temperature has been controlled around 25 degrees C. As I mentioned before, we use glycerol as a carbon source here, and then we use nitrogen source, which is nitrogen gas. And then I prepared a small video about the experimental setup in order to explain everything easier. This is my lab. And then we use 250 ml culture tube to cultivate the bacteria. As you can see, the bacteria is purple in color. And then that's with the specific bottle cap as well. It comes with liquid port and gas port, which is easy for gas and liquid extraction. We use the temperature controller together with a hot plate as well as an air conditioner in the room to maintain the temperature. And also the mixing is being provided by the internal magnetic flake together with the sterol. And then let's go back to the presentation. So Traditionally, like lots of people pointed out, for microorganisms, they grow really slow, and then the turnaround timing is very long. So that's why originally people would think you grow the bacteria and then they produce a hydrogen, so you harvest the hydrogen at the same time. So here are very preliminary results for the growth-associated hydrogen production, which is a very traditional method. So if you see the figure, and then you can see the empty diamonds represent for the cell concentration, and the solid square represent for the hydrogen production. So as the cell concentration grows, and then the hydrogen production grows as well. And then when the cell concentration stops to grow, and then it just doesn't produce hydrogen anymore. Also, on the other hand side, because we use glycerol as a carbon source and nitrogen and as a nitrogen source for the cell growth, you can see the similar trend as well in the lag phase, which is noted as stage 1 of this process. And then everything has been slow, and then in the exponential growth phase of the bacteria, and then the consumption rate of glycerol as well as nitrogen increase a lot. And when you utilize all the glycerol, the cells stop to grow, and then no hydrogen produced and no nitrogen has been consumed. Therefore, we can say that bacteria is very capable of growing on the glycerol and then convert glycerol into hydrogen as a side product under the nitrogen fixation condition. The good thing about it is that the hydrogen purity was very high, it's about 90 to 97%. But the downside of it is very slow process, like everyone points out. And also, the tricky part is that the glycerol to hydrogen conversion was really low. It's only about 80%. So you don't really use that much glycerol for producing hydrogen gas. And then if you want to understand a little bit more about where does the glycerol goes and also why the yield is so low, if you take a very close look about the nitrogen fixation process, you can see that the majority of the energy carrier, which is ATP, has been used to the ammonia synthesis, and also the majority of the electrons goes for the ammonia synthesis in that process as well, and that's for cell growth. But then if you think about, if you remove all the nitrogen sources of the whole reaction, you can see that you just use the electrons, which is a substrate, and also the energy comes from the light exclusively for hydrogen production only. So you don't waste anything else. That's by theory. How are we going to achieve that in the experiment setup? So we simply grow the bacteria like what we did before with glycerol and nitrogen gas, and then when it enters to the early stationary or later exponential phase of growth, and then we harvest it and then redilute it into with fresh media. And in that case, we only grow them with glycerol, so we don't put any nitrogen gas anymore, and hopefully they won't grow, and then they will give us some hydrogen gas out of it. And then this is a very preliminary result we've got for the non-growth associated hydrogen production. And so as you can see that in the first part of the timeline, there is still a little bit cell growth, which we suspect is because of the nitrogen left in the previous stage. But then afterwards, when it enters to the actual stationary phase of it, and the cell growth was controlled about like 4%, so it didn't grow that much. But then afterwards, that's a very interesting part coming out of it, and the hydrogen production rate, which is very constant, is a linear line, as you can see, and also from the glycerol consumption rate, that was linear line as well. So you get constant hydrogen production during the non-growth associated hydrogen production, which is very appealing and attractive in the industrial process. In the end, when you use all the glycerol, the cells start to perish, and then you don't produce any hydrogen afterwards. So the good thing about the non-growth associated hydrogen production is that you get similar hydrogen production rate, but you get similar purity. But the most important thing that you improve the glycerol to hydrogen conversion from 8% to roughly about 80%. So it's about 9 times more. And then that's why when you compare about the growth or non-growth associated hydrogen production in industry, it goes slightly favor to the non-growing hydrogen production, because we only use very small and light concentrated bacteria for this biological cultivation. But think about in industry, you can use concentrated non-growing cells and then immobilize it, use it just as a biohydrogen catalyst, and then produce bioconverted hydrogen. And then that will be very appealing in industry. Therefore, if you are doing experimental or small to intermediate size of biological cultivation, you can do the growth associated hydrogen production because it's easier to do it. But then if you are going to do more industrial scale and then try to make more benefit out of it, you should always consider about the non-growth associated hydrogen production. And also, here is a very simplified schematic plot about a possible example how to use that in the hydrogen production plant. Simply you grow the bacteria by using all the waste and solar power, and then harvest the cell mass, and then use that for the non-growing hydrogen production. Hydrogen will be utilized and also together with the oxygen from the air to produce electricity by application of hydrogen fuel cell. I have also prepared a very small video for a demonstration purpose. Hopefully that's going to be more interesting. So after the hydrogen has been produced from my prototype, it will be collected through the replacement for water in the burette. And then I normally use a syringe with a stopcock to extract the gas out. And on a daily basis, I simply send the gas samples to GC gas chromatography, so I can have a very clear idea what's inside and what's the gas composition. But in order to make it more interesting, I use a small hydrocar model to test the hydrogen of it. So simply just inject all the gas into it and see what's going on afterwards. Well, like you can see, I was a bit clumsy and worried about it. But then, yeah, but then after you connect everything, and then you just switch on and hope the miracle will come and it will work. Well, see, as you can see, the hydrogen, because I'm very sure what produced is very good quality because I did a gas chromatography test. That's why I can power this car. And this small hydro car contains a very small membrane hydrogen fuel cell. Basically, we'll utilize the hydrogen we produced and also the oxygen from the air to generate electricity and power the car. Obviously, that's enough for today, and I'm not going to show anymore. So let's go back to the presentation. And then a lot of people are questioning, oh, it's an interesting project, and it's fantastic, but then how much hydrogen you are going to produce and how you are going to use it in the daily life. And then lots of people don't have a kind of quantitative idea. So I did a very simplified calculation based on it. So if you have just about 1 kilogram of the bacteria and then just about 1.8 liter of glycerol, based on the very preliminary result, which we didn't do any optimization process in the past, and then now we'll produce about 114 liters of hydrogen per day continuously for a month's time. And then if you think about that much of hydrogen converted into energy, that's equivalent to that amount of energy. And then it could support all the house appliances running for quite a bit of time. just for every day, and then and also all these results are we got from the first year results, and now I'm doing my third year already. So we've done lots of optimization work, and then we can improve the result by about like 3-4 times. Sometimes if you do the cell immobilization work, it could be 10 times more hydrogen production, and depends on how you proceed it. If you're interested about it, you can always ask questions afterwards. And also due to the short notice, that's why I didn't put into this presentation. And then at this moment, I'm going to say a few words about people who actually make a great contribution to this project. First is the CAST Cambridge Research program for supporting the generous funding to this project, and also other professors and academia people for academic support. And there are a lot of people in our department who actually made lots of contribution on the technical side of it. And also I enjoy my friends accompanying my research group that are very supportive as well. Any questions?
Yes, we have a few minutes. Any questions?
So what is the yield of hydrogen as percentage of the theoretical maximum?
Yes, for the cross-associated, which is a traditional way to do it, is about like 10%. But for the non-growth associated, we can achieve it between 80% to 97%, depends on how you proceed it and what kind of concentration of bacteria you are using and what kind of method you are using. Yeah, if that answers your question.
Yeah. And what other products are there in addition to?
Yes.
So hydrogen is just a byproduct. What are the main products?
Okay. So traditionally, in the growth-associated hydrogen production, most of the material will goes for the biogrowth. So it's like a cell mass. It's a majority of the end product, and hydrogen is a side product. For the non-growth-associated hydrogen production, and then hydrogen, and it's about like I mentioned, the yield is a little bit high, but then that's just only on hydrogen part of it. If you do like a carbon balance of it, you can see that only a very small part of the carbon goes to the cell growth. The majority of the carbon goes to PHB, which is a polyhydroxybutyrate, a very good biopolymer, as you pointed out in your slides. Yeah, so if people are interested in any other biopolymer inside of it, then they can use that as well. And apart from the hydrogen product. So we have lots of any other refinery products we can generate here from this process. And also, people mentioned you can get 1,3-propanediol from this process as well, but not as much as a PHB though. Yeah.
We have time for one more question.
Thank you. Yeah, this work is quite promising and the process feasible, sustainable, but I'm not too sure about the interpretation of the process, if it's actually photosynthetic or chemosynthetic. We know that higher plants, for example, they have pigments, chlorophyll, xanthophyll, for trapping sunlight for photosynthesis. But when it comes to bacteria, except if this is different, Pseudomonas,
Yeah.
does it actually utilize solar energy for synthesis or chemicals?
Actually, so for the energy-wise, the bacteria will use, I can show you some results which I did in my later studies. Let's go down a little bit. So if you see the non-growing process, for the energy-wise, it will use some of the solar light, but then also it will use the reductive energy from the chemical side of it, which is a glycerol converted to anything else. And then that will trigger to the electron flow of the whole process. if that explains your question. Also, we did a little bit more research about what kind of spectrum of light they are going to absorb in the bacteria. If you see this diagram, this bacteria is a little bit different from any other green bacteria or any like an algae. These bacteria, they don't utilize water, they don't split water to generate hydrogen. They use organic substrate, and also the spectrum they are using is quite different. So like in algae area, it's more about visible light. Strong blue would be preferred, but then in these bacteria, they like more towards to the infrared or near infrared region. So if you are questioning about sustainability or anything else, and this bacteria can easily be co-cultured with a lot of other bacteria or algae together to use the whole spectrum of the solar light. That will make the whole process even more efficient, and it won't take that much water or solar light for single purpose, if I answer your question. Yeah.
Right. Thank you.
Thank you.
We have